Hey everybody, welcome back to our continuing discussion of neurochemical communication. This time we're talking about exocytosis and signal termination. Okay, so to talk about vesicular exocytosis, we need to talk about vesicles, right? So vesicles are these little sacs that float around in your terminal buttons. You can see they're composed of a lipid bilayer membrane, much like the membranes of your neurons. But instead of a neuron, inside of here we have little neurotransmitter molecules. In this example, we have glutamate molecules. So we have a lipid bilayer membrane, and inside of that we have neurotransmitters that can be released. In addition to those things, we have a number of proteins that are attached to or embedded within the lipid bilayer membrane. These include things like vesicular transporters that allow neurotransmitters to be inserted into the vesicle, and proteins on the outside that are important for things like docking and priming, which we'll talk about in just a moment. Okay, so basically, here's what's happening with vesicular exocytosis, or the process by which those vesicles fuse with our neurons' membranes and then open their contents to allow neurotransmitters to cross the synaptic cleft. We need vesicles to dock with the cell membrane in the active zone and prime to be released. So the active zone is this portion of the membrane here where there's sort of a density of locations for these vesicles to bind and release. There's also a large amount of calcium channels, which we'll talk about in just a minute. There are various proteins that are necessary for this step, which we'll show in more detail on a figure in just a moment, including some of those proteins we talked about, which are part of the vesicle membrane. So here in our active zone, we have voltage-gated calcium channels. Those are what they sound like. They are ion channels that are open in the presence of a sufficient change in voltage. So when the action potential, which we talked about last time, arrives in the terminal, it's going to cause a depolarization event, which is going to cause these voltage-gated calcium channels to open. That influx of calcium will cause vesicles to fuse with the membrane of the cell and release the neurotransmitters into the synapse. And as we mentioned already, calcium channels are concentrated here within the active zones of the terminal membrane, the places where vesicles can fuse and release the neurotransmitters. Okay, so to go through that with this image here from your textbook, the action potential invades the terminal button. We have depolarization, which will cause the influx of calcium ions through these ion channels. So the calcium coming in will then cause these vesicles to release their neurotransmitters into the synaptic cleft. After they're released, the neurotransmitters will bind to receptors on the postsynaptic membrane, which will cause the opening and closing of postsynaptic channels, which will then cause changes in the postsynaptic current. So the excitability of this postsynaptic cell changes. So that's the complete picture. That's how neurons talk to one another, right? When this action potential invades the terminal button, it causes, through the influx of calcium and the docking, priming, and exocytosis of these vesicles, neurotransmitters to be released across the synapse, which will bind to receptors on the postsynaptic membrane, which will cause ions to flow through here, causing changes in this postsynaptic neuron, EPSPs and IPSPs, right? All the stuff we talked about last time. So after that happens, the vesicular membrane is retrieved from the plasma membrane and can be used again. We'll talk in a bit about what happens to these neurotransmitters out here after they've had a chance to bind and do their. So as we mentioned, during exocytosis, vesicle membranes will join with the cell membrane, right? They're all made, both of these membranes are made out of the same sort of lipid bilayer. So during exocytosis, the cell membrane can join with, the vesicular membrane can join with the cell membrane. This vesicular membrane can then be recycled via endocytosis, in which the cell membrane buds and pinches off, which fuses with an endosome. An endosome is just a membranous compartment within the side of the terminal. This little membrane here is going to help the vesicle bud off and be recycled as a vesicle once again. Okay, so let's go back and look at this in a little bit larger detail. So on our vesicles, we have a cluster of proteins that are embedded in the membrane of the synaptic vesicle, which can match on here to these clusters of proteins that are part of the synaptic membrane. This attachment here between these groups of protein clusters is what we call docking and priming. So once that's happened, our vesicles are docked and primed, ready to release their neurotransmitters. All we're waiting on is the influx of calcium. So when calcium enters the presynaptic membrane, right, we're in the active zone here, so we have all of these calcium channels, voltage-gated calcium channels. So when the action potential arrives, it's going to cause calcium to flow in through those voltage-gated calcium channels. That's going to facilitate the fusion process. So once the calcium is in, it's going to help these duct and primed vesicles fuse. When they fuse, we have what's called an omega fissure here based on the shape. The neurotransmitters are going to be released out of this compartment, diffuse across the synapse, and do their thing. After that, this budding process begins, and with the help of an endosome, the vesicle is sort of taken out of the membrane of the neuron and can be recycled, repackaged with neurotransmitters, and used again. So the mechanism of exocytosis can also be a target for drugs or toxins. For example, botulism toxin blocks neurotransmitter release at neuromuscular junctions. So botulism toxin can be found sometimes in improperly preserved, like canned or pickled foods, and it's extremely deadly. It's extremely deadly because it blocks the release of acetylcholine at the neuromuscular junction, leading to paralysis. And severe paralysis can cause respiratory depression and death. So oftentimes our neurons will recycle neurotransmitters after they've crossed the synapse and done their thing. There are things like reuptake mechanisms which will take those neurotransmitters and put them back into the releasing cell, allowing them to be repackaged and used once again. This only happens with small vesicles that contain classical neurotransmitters, such as amino acids or monoamine. Neuropeptides, however, can't undergo this process because neuropeptides are derived from precursor proteins that must be packaged into large vesicles in the cell body. Because of those properties, recycling of these vesicles can't happen at the terminal, and these neuropeptides are typically broken down with enzymes after they've done their thing. So as I touched on a little bit earlier, there also exist what we call retrograde messengers. These include gaseous and lipid neurotransmitters, as you can see here in this figure I've taken from the textbook. So lipids and gaseous neurotransmitters pass through membranes, and they can't be stored in synaptic vesicles, right? They're just too permeable. They won't stay put. So as a result of that, they are made on demand and then can diffuse out. Once they're in the extracellular fluid, they can travel to cells that are far away or immediately in the vicinity. So typically you see release of retrograde messengers by the presynaptic cell as a way to sort of signal backwards, right? Retrograde, they'll diffuse back across the synaptic cleft and have their effect that way. Though many of these do function outside of the central nervous system, such as nitric oxide. So in this example, acetylcholine can cause an increase in the synthesis and release of nitric oxide, which serves to promote vasodilation, or the dilation of blood vessels in the periphery. So how this might look in terms of a larger scheme is we have our presynaptic cell signaling onto the postsynaptic cell through a classical mechanism. So we have neurotransmitters and vesicles here being released, which will generate an intracellular signal. We're being kind of vague here because we're talking about, in general, gaseous and lipid neurotransmitters. Of course, each neurotransmitter is going to have its own intracellular pathway, which will increase its synthesis and release. But sort of generically speaking here, neurotransmitters stimulating an intracellular signal. That signal is going to cause the creation, the synthesis of retrograde messengers, like lipid or gaseous molecules, which can then diffuse freely through the cell membrane and travel backward to influence the presynaptic cell. So neurotransmitter release can be regulated in a number of different ways. So here we have pictured our presynaptic and postsynaptic cell and sort of zoomed in on sites of transmission that we're going to be talking about. So release is regulated by a number of different factors. First is the rate of neuron firing. So as we talked about, the action potential reaching the terminal button is going to allow that calcium to come in and cause the release of neurotransmitters from the vesicles via exocytosis. Generally speaking, a higher rate of neuron firing is going to mean a higher rate of neurotransmitter release. But there are other phenomena that can interact with this. We'll talk later on about how some receptors can become desensitized if they're overstimulated, which could lead to decreased signaling. And there's also the potential for large neuropeptide neurotransmitters to be depleted with heavy rates of fire. Likewise, the probability of vesicles undergoing exocytosis can be affected by a number of different factors. For example, we talked about botulinum toxin, which will prevent the release of neurotransmitter from vesicles by interfering with that process. The thing we're going to spend more time talking about is the influence of autoreceptors. Autoreceptor, right? Auto being a prefix Greek for self, and receptor being receptor. self-receptor, right? This is receptors for the same transmitter released by the neuron. So if a neurotransmitter releases acetylcholine, for example, an autoreceptor would also be sensitive to acetylcholine. It's a receptor located on the releasing neuron that is sensitive to the neurotransmitter that neuron releases. So it's a way of detecting what is being released by that neuron. It's sensitive to the neurotransmitter that neuron releases. As you can tell from this figure here, we're going to talk about two different places we can find autoreceptors. The first are terminal autoreceptors. These are located on the presynaptic terminal, right? So this is the presynaptic terminal that's releasing neurotransmitters across the synapse. So we can see that the neurotransmitter is released and then makes its way back around here to the autoreceptor and binds and produces its effect there. It's activated by the released neurotransmitter, as I've said a few times already, and typically this is going to serve to inhibit further neurotransmitter release. So you can see here, this is indicating through sort of a vague mechanism. Like most things we talk about, the exact mechanism that this is going to operate through is going to vary from neuron to neuron or neurotransmitter system to neurotransmitter system. So we're going to have, generally speaking, a decrease in the amount of neurotransmitter that's released if the terminal autoreceptor is stimulated. And here, sort of by contrast, we have the somatodendritic autoreceptors, which, as the name suggests, they are found on the soma and dendrites, and they are autoreceptors. So they will function through a slightly different mechanism. They slow the rate of neural firing, generally speaking, which will decrease the rate of action potentials, which will decrease the rate of neurotransmitter release, right? Fewer action potentials arriving in the terminal means less calcium coming in, which means less vesicular binding and release. Lastly, we'll mention that some drugs can block or act on autoreceptors. We'll talk about some examples of these later on in the course. Okay, here's another more visual way of looking at how autoreceptors might work. We have our presynaptic neuron here in green and our postsynaptic neuron here in blue. When this vesicle binds and releases its neurotransmitters, you see here that one of those neurotransmitters makes its way over to this terminal autoreceptor. While these neurotransmitters will function normally and allow the passage of ions into this postsynaptic neuron in this example, the function of this autoreceptor is going to decrease the amount of neurotransmitter released in future binding events. I'll also mention briefly here heteroreceptors. These are receptors that regulate the synthesis and or the release of neurotransmitters other than its own ligand at the axoaxonal synapse. So here we've shown an axoaxonal synapse. We've looked at one of these already. We've talked about facilitation and inhibition at the terminal. And this is basically the mechanism that this operates through. So we have here this neuron influencing the terminal of this neuron over here, either enhancing or inhibiting neurotransmitter release happening at this synapse in here. So it works kind of like an autoreceptor might, except it's not an autoreceptor. It's a heteroreceptor, right? Because it's not on itself, it's on a different neuron. So it is sort of functionally adjacent in that it is stimulating a receptor on the terminal, but it is not the type of neurotransmitter that is released by this terminal. Okay, we've talked about vesicular storage and release and what happens when neurotransmitters cross and bind and do their thing, but signals have to be terminated, right? It would be kind of a bad situation if a neurotransmitter was released and just sort of bounced around in the synapse, binding over and over and over again. So neurotransmitter molecules have to be removed from the synaptic cleft. We're going to talk about a couple of different ways that can happen. The first we're going to talk about is enzymatic degradation, which is what it sounds like. An enzyme, which can deactivate that specific type of neurotransmitter, will come along and inactivate it, break it down into its constituent parts. This is a common end of the signal for non-traditional neurotransmitters, things like acetylcholine, retrograde messengers, and neuropeptides. These molecules that cannot easily be picked up and put back into the cell to be reused are degraded by enzymes in the synapse, and that's the end of their signaling career, right? Their signal is terminated as the enzymes take them apart. Later on, we'll talk about certain classes of drugs that can interfere with this process. So, for example, an acetylcholine esterase inhibitor will inhibit acetylcholine esterase, which is an enzyme that takes apart acetylcholine. And if you inhibit acetylcholine esterase, it's going to increase the amount of acetylcholine that's available, right? If you get rid of the thing that breaks something down, then that thing is not going to be broken down and we're going to have more of it. So a way to increase the amount of acetylcholine available in the synapse is to disable the enzyme that normally inactivates it. So another way that we can terminate the signal is reuptake. So some neurotransmitters can be taken up by the presynaptic cell or by astrocytes. We have transporter proteins like this right here that sit in the cell membranes that can help take up neurotransmitters after they've signaled. This is especially important for things like amino acid neurotransmitters. This process of reuptake will allow these neurotransmitters to be taken up from the releasing cell, or as I mentioned, by neighboring astrocytes, and repackage and reuse them. So perhaps unsurprisingly, there also exist drugs that work on these transporter proteins. For example, SSRIs, selective serotonin reuptake inhibitors, will prevent the reuptake of serotonin. If serotonin is not being taken out of the synapse by those transporters, it's going to continue to float around in the synapse and bind to serotonin receptors, magnifying the effect of serotonin by allowing it to stay out in the synapse for longer. Okay, that's it for vesicular release and signal termination. Next time we're going to move on to talking about receptors.